Hi, I'm Dhavi, a product manager at Google DeepMind covering genomics. I'm joined by Ziga, who leads our genomics team, Natasha, Tom, and June. We've just released our recent work, AlphaGenome, in Nature. AlphaGenome is a unified DNA sequence-to-function prediction model that predicts the functional impact of genetic variants. And today we wanted to talk about the science behind it, why we built it, how it came about, what we released, and what we're excited about next. Maybe the best place to start is, why did we build AlphaGenome? Ziga, how does this fit in with the overall mission of the team?
So the mission of the team is to build an AI system that will allow us to decipher the genome sequence. The genome, or the DNA, is the source code that the evolution has programmed across millions of years and for millions of species. So it's the source code of life. And deciphering this source code would have enormous benefit in health and many other areas. And one way to measure our progress towards this goal is to look at how well we can predict the effects of mutations or small changes to the DNA on molecular mechanisms of the cell that read this DNA.
And why is this important, AI that helps us understand and decipher the genome?
Yeah, I think improving our understanding of the genome has such a profound impact on everyone's life. If you think about all the people suffering from genetic disease, and in fact, a large fraction of the rare genetic disease remain undiagnosed today. So AI is actually we have so poor understanding of the genome as a whole, and AI is such a good tool to read the genome and make meaningful predictions about the genetic mutations. For me, I think understanding the genome and predicting mutations is one of the most impactful applications of AI can make.
How did you get interested in this field?
I started working on predicting genetic mutation during my PhD. And this has been my interest ever since. I think, when I joined DeepMind, I was leading AlphaMission's project, which was a method to predicting the genetic effects from mutations from the coding region, which was not only 2% of the genome, but what about the rest of the 98% of the genome? That's why we're presenting AlphaGenome today. And this method is focusing on non-coding part of the genome, and hopefully shed some light on the the majority part of the genome. And also, speaking a bit more personally, the reason I got into biology is because when I was at high school, I was reading a college textbook. It was describing all these fantastic and complex, elegant molecular mechanism of life. That's why I decided I should work on biology for my career. And Tom, what's your experience there?
It's quite great to have such a multidisciplinary team. We all come from different walks of life and bring different things to the table. So I think for me, and I think other people who hadn't worked in biology, I think genetics is a really fascinating field. It seems like such an unexplored problem space, and being able to work out what very small changes in DNA can cause in humans and other species is really quite fascinating. And the field is, especially in machine learning, the field in computational biology seems to be kind of going really, really fast, and there's lots of things going on in that field. So it's a really exciting place to work.
I can still remember, before I joined DeepMind, I worked on BPNet, which was a tiny network in today's standards. It took as input 1,000 base pairs of the DNA and predicted transcription factor binding at very high resolution. And I was fascinated by how rich and sort of precise this information can be at base resolution. But at the same time, it felt like very much a zoomed-in picture of a specific process. So when I joined DeepMind, I wanted to work on sort of a more complex processes. So we worked together with David Kelly at Calico on predicting gene expression from DNA sequence, which is a very difficult problem. And for that problem, we had to extend the length of the sequence quite significantly in order to capture as many relevant pieces as possible. But in order to do that, we had to trade off the resolution that made BPNet so interesting. So it's always felt like a bit of a trade-off between these two things, the sequence length and the resolution.
I guess there have been many other models pushing this area forward. What was the real gap that we were trying to fill with AlphaGenome?
There have been other DNA sequence to function models before. The most similar to AlphaGenome is probably Informer, also from this team, and Borzoi from David Kelly's group in Calico. And there's also lots of more specialized single-task models. There's Splice AI for splice size. There's, Orca and Akita for contact maps, ChromeBPNet for accessibility, and so on. I think the gap that AlphaGenome has filled is it's modeling more modalities in one model than ever before. The input sequence length is very long, and the predictions are very finely detailed. So I think it's a really powerful combination of these three things, the modality coverage, megabase of DNA sequence, and single base resolution outputs for many of the output types. I think that makes the model quite useful for variant effect prediction and variant interpretation. And of course, also the increased predictive kind of accuracy across all these different outputs. The utility and convenience point, I think, is very powerful, actually. So now with one model, you can kind of look at the effect of a variant from many different lenses and consider the effects on different layers of molecular biology, whereas before you might have had to use many different models to do that analysis.
Why did we need separate models? Why is it so hard to do long context, high resolution, multimodalities all at the same time?
DNA sequence to function models work by basically taking a chunk of DNA and getting the model to predict real-world experimental data. And so like you said, like the complexity comes from when you want to make kind of more longer range sequence predictions to higher resolution and with lots of different modalities, you kind of run up against these kind of computational limits and like high memory usage, which makes it very kind of difficult from an engineering perspective. And so for AlphaGenome, we were really keen to try and break that, those trade-offs and try and do kind of all of those things in a single model.
And how did you really get around that problem then?
Yeah, we ended up taking the sequence and splicing it into different subsequences and then using multiple TPUs to process these subsequences, which are then much shorter in length. And that allowed us to use much less memory on a single TPU. The key piece we had to make sure is that they still communicate with each other so that the TPU1 can still communicate to the last TPU in sequence. And so instead of sampling intervals in the genome randomly, we sampled them next to each other and then allowed these TPUs to talk to each other. I can still remember the day when we were having lunch with my colleague Guido, and I shared this idea with him, and we started discussing. And at the end of the lunch, we weren't really sure if it's going to work. A few days later, he comes to me and says, I have a prototype running with fake data, and it runs almost as fast as our Informer training loop, and it can process these very long sequences at high resolution. And at that moment, I was really excited because I knew that, this can theoretically break this limit of long sequence and high resolution. Mm-hmm. Although in practice, it turned out that we were still quite far away from making it actually work, right, Tom?
Yeah, so once we got the model parallelization working, the next thing we needed to do was be able to feed this model with vast amounts of training data. I think at one point we were loading kind of 40 or 50 gigabytes of data to try and keep up with the model's training. So we had kind of two big realizations. One was that the data is actually really sparse. It's like kind of 99% zeros in some of the modalities that we were training for. So that meant that actually all we really needed to focus on was being able to compress and decompress the data as fast as possible. And so by doing that, we managed to kind of break that loading the data fast enough for the model to be able to kind of feed it and train. And then the second thing we did was we were really ruthless on the data that we were feeding the model to train. We did kind of pass a couple of quality check passes to make sure that the data was either really high quality or it was added to the diversity of the model so that it could get train, generalize a bit more or learn new capabilities. So once we'd kind of got the data loading working, we were able to then train the model really efficiently and be able to kind of iterate on the modeling as much as possible. And so we'd kind of got the 1D representation of data training and kind of predicting. And so the next thing we were really excited about working on was kind of two-dimensional data, so content maps and splicing. So Jun, I think you worked quite a lot on the splicing. It was definitely a very tricky problem.
Right. That was quite tricky. So splicing is a process that is needed for a gene to express a protein properly. And, it's a process, the genetic information encoded on the genome non-contiguously. So if you want to express a functional protein, you need to bring all this non-contiguous information together, and that process is called splicing. As you can imagine, this process can easily go wrong due to genetic mutations. And in fact, many genetic diseases are caused because, splicing defect. I worked on this problem during my PhD, and the method published then was called MS-Splice. We're trying to model the competition between junctions, but the model wasn't nearly as good. So I had these ideas how to do this properly back in my mind, and when we started working on AlphaGenome, I realized that this is a perfect natural fit, and with the talent team, Guido, Tom, and others, we finally have all the technical pieces together to properly do this modeling work. And speaking of that, it wasn't really that easy as it sounds, because the output format and input format was quite different, and we were predicting is 2D extremely sparse arrays instead of 1D track. And in the end, we finally got it to work, and I'm really, really happy with that. And this kind of new capability is actually new to the field. It's definitely also new to the team. And after we get this part work, and the next big part is about content map. And Natasha, how we made that work?
Yeah. Contact maps. So contact maps are two-dimensional matrices that capture interactions between DNA regions. And it turns out these interactions are very important for gene regulation, essentially. So the genome in the nucleus is folded in these intricate three-dimensional shapes, and the proximity between regions is really important for things like promoter-enhancer interactions. These are two types of regulatory regions which can massively impact a gene's expression. And then there's also insulator elements that specifically prevent these interactions. And so these are fundamental biological processes, and it's a bit of biology that we definitely wanted in the model. Luckily we managed to add it in, get it training end to end, and actually we were happy to see that adding this modality didn't lead to any degradation on the other modalities we already had.
Right, it was quite interesting to see that how we were able to add these modalities, both 1D modalities as well as then 2D modalities like splicing and contact maps, without hurting the model's performance or slowing it down significantly. In the hindsight, it kind of makes sense, right? Because all of these modalities or measurements are measuring the same underlying process, like transcription or splicing. However, it's still nice that this worked out.
And as you add all these new modalities, how do you check it's not hurting performance? How do you evaluate it?
Our evaluation strategy was kind of in two halves, two-pronged. The first is checking how well does the model do on new DNA sequences that it's never seen before. How accurately can it predict all these different modalities, like all these different signals along the DNA sequence? The second half, which we think is more interesting, certainly for downstream applications, is again variant effect prediction. So given a small mutation in the DNA sequence, this might cause downstream changes, and how well can the model recapitulate that? The way that we do this is basically we have two DNA strings, one with a mutation and one without. So just the reference genome sequence. We feed that into the model separately. We get predictions out, and then we basically look for differences. We look for differences and we summarize them. So conceptually, at least, it's fairly simple, but technically this caused like endless headaches, essentially. The first thing that we ran into was speed, actually. So the model outputs are something like 11 gigabytes, and variance scoring was notoriously slow.
So very much in the same vein that we had with feeding the model data, the next one we had was basically trying to get these large predictions back and kind of summarize them into kind of useful like statistics that we could use to kind of evaluate how well it's doing on a particular task. And so we kind of realized that everyone on the team was kind of doing it in their own little way. They were like coming up with their own aggregation strategies and doing it in like like scripted on notebooks. But it was, like you said, really slow. It'd take sometimes minutes to make these predictions. And so a few engineers kind of sat down and we kind of worked out how to kind of create this kind of variant scoring API, where we could make these variant predictions and aggregate them on the same devices that we were running the model. And that meant that we could do it like really fast and really efficiently and like doing lots of different aggregations in parallel. And that allowed us to do kind of, lots of evaluations really, really fast. And we did quite a few evaluations for the paper.
Yeah, lots of evaluations and benchmarks and evaluations stuffed into the paper. But I think the reason we did that is we wanted to evaluate the model as comprehensively as possible, really, given constraints of time and space and sanity. One issue that we ran into very early on, actually, was how are we going to structure our evaluation strategy? Like, how do we make sure that we're doing it in a rigorous, comprehensive way? Because the model does have so many capabilities, how do we handle all of that? And the approach that we took on was, again, parallelization. And so essentially what happened is each individual would take on one or two areas, like maybe one or two modalities, and kind of own that section of the paper really end to end. So they would ingest the data, design the evaluation, add benchmarks, make the paper figures. And this worked really well, actually. And the other thing is that a lot of these evaluations were already pre-existing in the research community. So in a lot of cases, we could just ingest them and use them directly. So that was extremely helpful.
You mentioned different people started focusing on different areas. Are there different classes of evaluation then?
June can talk to that.
Yeah, exactly. Whenever possible, we try to evaluate our model in different ways. For instance, we evaluate on the molecular level if we recapitulate the experimental readout well, and also on the organism level consequence, if we can predict well a mutation is going to lead to higher disease risk or not. Also, we also try to show as many examples as possible, like both examples where the model did well and examples model did not well. And this was just one example I found particularly interesting that we showed in the paper. This, the model was trying to predict some cancer driver mutations, and we were very surprised to find that the model actually did very well recapitulate what the researchers found in the wet lab, and that was amazing to see that the model ranks the driver mutations much higher than some other random controls. We are quite happy and put in the paper.
Awesome. Yeah, I guess after the evaluations, it was kind of down to the paper writing.
Yes, just the paper writing.
Yeah, why is the paper so long? It's practically a novel.
Yes, it's so long. But I think inevitably it was always going to be long, just because the scope is massive, essentially, or at least I think it is. We really wanted to do justice to all of the other models that we compare against. We really wanted to dive as deeply as we could into all the different modalities. So it is what it is. And I think we also suffered a bit from one more eval syndrome. We're all very excited in this field, and people would go off and see like a new paper or a new data set, and they'd want to integrate into the model and add just one more eval, one more figure. But I do think the paper is a lot stronger for it. So I'm happy that happened.
Yeah. And how long did this whole process take, I guess, from that initial prototype to having the paper?
I think that the team has been working in this area for quite a while, but if we were to pinpoint that particular lunch conversation and to when the paper was published, it was under 2 years. I think the team worked really hard to get to that point. At some point, we all got into one room, put a lot of monitors on the table, and we worked together on this. It was a very exciting time to do that and to see the progress. And I'm really excited and very proud of the team, how we came together and were able to release the model and the API.
So I joined the team when the model was almost ready, and we were starting to think about how to share this with the community. And there was a real eagerness to both share something quickly, but also try to find a way to make it easier to use.
Yeah, we were really quite pleased with how we were built this kind of model API for interacting with our model and making predictions and kind of evaluating and analyzing the results. And I think we quite quickly realized that we wanted to make the same thing available to people externally as well, in as easy and accessible way as possible, whilst also keeping the versatility of being able to do lots of different things. So I think one of the cool things we did was basically making this API available at the same time as our preprint. And the cool thing was that you can kind of just open up a notebook and make a prediction and visualize the results really quickly without the need of GPU or installing drivers and all the other kind of faff that comes along with loading a model from scratch. I think that was really powerful. So we were keen to do that and see what how people used it.
Mm-hmm. With things like making it simple and easy to use, something that we spend a lot of time talking about as a team is also what is this actually allowing people to do at the end of the day? Jun, what are you most excited about?
Yeah, there's a couple of things I'm quite excited about to see, how the community use AlphaGenome. I think the first is as a tool to help scientists to pinpoint what kind of mutations could be, harmful to human. Another is use this as a tool to understand basic biology, right? If you think about biology is quite complex, and with this tool, hopefully, scientists can start to study, make predictions about which part of the genome is functional and which part of the genome is responsible for regulating gene expression and which part is activated in which cell type, things like that. I think, this is be a fantastic tool for that purpose. With AlphaGenome, hopefully scientists can, start using that and to maximize the return with the limited resource and funding they have, and to basically improve scientific research.
Ziga, Natasha, you've both been in this field for a while now. What are some of the requests we've gotten, and has there been anything surprising here?
Yeah, people had lots of, quite a few feature requests. One of the ones where, for example, people are asking, can they do not just single letter changes to the genome, but multi-letter changes like insertions or deletions or large structural variants? And for Indel's, actually, this is a feature we already had it implemented. It just wasn't highlighted in our quick start enough. Although one user did find a bug in this code, which is actually quite tricky, and we managed to fix it. And the other feature was, people were asking, can they use the embeddings of the model, or can they fine-tune it on their own data? Because there's quite a lot of more interesting data sets that we weren't able to include in our model, right, Natasha?
Yeah, actually, on the data front, I was intrigued and maybe not surprised by the amount of requests for additional species, additional modalities, and additional cell types. So as we've sort of mentioned already, we've tried to make the model, have reasonably good coverage, although not on species. That's still just human and mouse for now. But basically, people want more capabilities under one roof, and that makes sense. Actually, I can definitely relate to that. One thing I wanted to highlight is that this type of feedback signal from the community is incredibly important to us. It directly feeds into what we work on next and what we build next.
Now for a question we get asked all the time, what's next?
Yeah, and if you go to the website and look at the API, we're predicting tens of thousands scores per variant, which is obviously very hard for scientists to read and reason. So we're thinking how we can summarize all these scores to a single score per variant, and basically aggregate the scores as much as possible, so that users or scientists can make prediction per variant and look at the scores and then pick prioritize a short list of variants they want to dive deep into. And then they can look at the very comprehensive predictions per cell types and tissues, and then, answer the questions that which effect this variant come from, which tissue the effect come from, and what genes activated, things like that.
Yeah, then the other thing we wanted to do is support kind of really large-scale analysis, like genome-wide association studies and things like that. Currently, the API, you can make many lots of predictions, but maybe not quite as many as those kind of like really wide, large scale kind of studies would need. So yeah, we're looking at like basically trying to maybe pre-compute as many variants as possible, so that people can do those large scale analysis much more easily.
And we're releasing the model weights as well, of course. Yeah, we're really excited to see what people do with those. So that can be anything from playing with the embeddings or fine-tuning or extending with new data. And because the model is good at so many tasks, we're just really excited to see what people use it for. Hopefully it can be applicable to many different diverse problems, some of which we've probably never even thought of before.
I'm so happy we're going for all of this, and hopefully hearing it makes some of our scientists happy as well. Ziga, maybe going back to the team's mission and how AlphaGenome was sort of a step towards it, how are you thinking about what's next for the model?
Yeah, I think there's still a lot of headroom for improvement, like a lot, both in terms of predictive performance as well as capabilities. In terms of predictive performance, we'll see how far we get. And in terms of capabilities, one of the things I'm excited about is to leverage these new amazing single-cell atlases, such as because the technology has been really evolving quickly and improving. And this allows us to measure not just the molecular processes at the tissue level, but at the individual cell type level within the tissue. And if we could extend the model with these, then I think it would be more applicable to studying variants associated with diseases where, for example, this sort of a defect arises in these individual cell types.
Thanks for watching. Those were some of our highlights on building AlphaGenome and releasing it. There's a lot more details in our Nature paper, and we'd really love to hear from you as you start exploring and using the model, whether that's interesting case studies or gaps or ideas that can make the work more useful for you. Our user support team is basically us, so please get in touch with us on the Discourse Forum.